Hello, everyone. Thank you all for joining us today. My name is Claudia Labrador. I am the Global Product Manager of Reagents, Kits, and Assays at Stilla Technologies, and I will be the moderator of today's session. I am excited to introduce our expert speaker, Sahbi Ben Kilani, our Global Field Application Scientist Manager, who brings a wealth of knowledge and hands-on experience in the field of molecular biology, providing extensive support to digital PCR users. Sahbi played a key role in launching Stilla's innovative 6-color solution in 2021 and just recently oversaw the launch of the 7-color Nio Digital PCR, as well as Crystal Digital PCR assays. With 12 years of experience in the digital PCR field, Sahbi has contributed a wide range of applications, including mutation detection, CMV characterization, and pathogen detection across various fields such as oncology and cell and gene therapy. He will be guiding us through today's webinar, feedback from liquid biopsy laboratories using pan-mutation panels for precision analysis with Crystal Digital PCR, where we will explore powerful applications of digital PCR and liquid biopsy analysis. Throughout this session, we'll be conducting a few polls related to digital PCR and liquid biopsy. We'll share the results at the conclusion of the webinar. With that, I'll hand it over to Sahbi.
Thank you very much, Claudia. I'm happy to present this webinar today. So I'll start sharing the presentation. Hope you all are seeing my screen now. So thank you very much. I'm really happy to present this session and to also share with you the feedbacks and the results from our users that think about their point of view about the next generation of digital PCR and how at Stilla we think that next generation GPCR will be in the coming years. Of course, at Stilla, we started developing digital PCR and working on digital PCR since more than 10 years. And we dedicated our development to innovate and bring innovative solution to laboratories with an easy and accessible workflow and systems. So we were the first to launch the 3-color system and also the 6-color system some years ago. Last year, we also launched the Nio system with the 7-color all-in-one system. And we also, just before that, launched the Ruby chip, which was also a very innovative consumable for digital PCR, as you will see during the presentation. And more recently, we also launched the new Crystal Flex assays that will be presented during this presentation. Since we started developing digital PCR, one of our first customers were working on liquid biopsy, and we supported them across all those years. And we also worked with them to overcome some challenges that we know that are present for liquid biopsy. As for example, when we work with liquid biopsy samples, there are many analytes that can be detected in this kind of sample. And we developed a methodology that can address those multiple targets like methylation, cell-free DNA, cell-free RNA. The other challenge is the concentration of the cell-free DNA, and more precisely, for example, in oncology, the circulating tumor DNA, which we know that the concentration is too variable, depends on the sample condition. And also this impacts the mutation frequency, which led to have a mutation frequency range between 0.1 and 10%. And this is why also we developed a technology that provides very high sensitivity to address this low mutation frequency. And also the last challenge is that the mutation coverage, because as you know, when we work with liquid biopsy, the volume of sample is too precious, and we need to have the maximum information from the sample to avoid wasting the volume and be able to use it for other applications. This is why at Stilla we developed, and also we are proposing this solution to have a fast screening of target mutation for different kind of diseases. But for today, we will only focus on oncology as example, and to show you some panels that cover multiple gene mutations. To show you also this high sensitivity detection that we can reach up to 0.1% of rare mutation with a very competitive price per sample and with the highest multiplexing mutation panel. And of course, all this with an easy workflow for routine testing and with one instrument, one consumable, and one pipetting step. To illustrate that, I want to start with an example of the usage of our solution in one laboratory that developed some panels for oncology for different cancer diseases with multiple panels to use with digital PCR at first instance instead of NGS. So basically, with those panels, the customer can screen those most common or more frequent mutations like EGFR, KRAS, BRAF, or ECR1 with each digital PCR at first instance. And if the result is positive, they can directly start their decision strategy. If the result is negative, they can try another specific assay for rare mutation frequency or other panels to explore other biomarkers. This strategy can be used for mutation, but as you can see, it could be also used for other biomarkers like methylation, which is something that we are more and more seeing in the field. And we have different biomarkers that we can detect with digital PCR, again, in first instance before going to other methodologies. So there is two strategies to use our technology for. The first one is when we have a lot of sample to test with a known panel, like for example ECR1, where we want to screen a lot of samples and for the same panel. This is the first strategy. The second strategy is what we call a profiling strategy, where we can go up to more than 100 multiplex panels to explore many biomarkers at the same time. So it could be mutation panels, CNV panels, methylation panels, or other at the same time in the same consumable. And this is why at Stilla, we believe that this, and also our customer also think that this is the next generation digital PCR usage. So if you want to position Stilla technology in the molecular biology market, we will be very close to targeted NGS than the basic quantification digital PCR platforms. To show you some example of this usage of digital PCR with those multiplex panel, I will show you this first example of the BRAF, KRAS, NRAS assay that was developed for one of our customer to target different kind of cancers. So we try to evaluate the robustness and the sensitivity of these assays with our technology by running two experiments. The first with a sample that we know that the mutation rate is about 2%, and we done a serial dilution of the sample to be in the liquid biopsy concentration range. The second experiment is that we diluted the mutation in the wild type to achieve an 0.05% of mutation frequency. So this will show us the level of the sensitivity that we can reach. So as you can see here, those are the data that comes from this assay in our software. And as you can see, it's very easy to interpret. As in the first example here, we can see the mutation of BRAF in this yellow polygon. We can see the mutations from KRAS in this second blue polygon, and the mutation from NRAS in this green polygon. So as you can see, in terms of visualization, it's very easy to interpret. And of course, from this visualization, we can then extract the concentration and the mutation rate. And here are the data of this first experiment of diluting the sample with 2% of mutation, starting from 6.5 nanogram per microliter to 0.4 nanogram per microliter. And as you can see, the mutation range is always around 2%. And of course, you can see the good linearity of the dilution for the wild type, but also for the mutation concentration. And as you can see, this is for the 3 genes. So just to make a quick reminder, this is a 6-plex assay. So it's covering all the mutations that are in the hotspots of each gene. The second experiment where we tested the sensitivity, as you know, liquid biopsy sample concentration is very low and with a range which is between 3 and 0.5 nanogram per microliter. So what we tested, we tried 3 concentrations. The first one is 1.7 nanogram per microliter. The second is 0.8 nanogram per microliter. And the third one, 0.4 nanogram per microliter. And for each concentration, we diluted the mutation in the wild type sample to decrease the mutation percentage up to 0.05%. And of course, we tested the wild type sample. And as for NGS, when we want to go in terms of sensitivity, we want to increase the sensitivity and the depth of the sensitivity, we increase the number of reads. Here, we increase the input volume to be able to achieve a good precision and sensitivity level. And here are the data. So as you can see, for each concentration and for each gene, we were able to detect all the mutation frequencies for each target. And as you can see, even for the very low concentration condition, we were able to have the right data and the expected data compared to the other concentration. So this shows that whatever the concentration of the sample, with our Ruby chip and our assay, we can achieve great sensitivity up to 0.5% of the mutated allele with the wild type, with a very good reproducibility and even in the very low concentrated sample. Now, I will introduce a new concept that we are using at Stilla and we are developing at Stilla, which is called the color combination, because for the next example, I will show you an assay that can detect up to 18 targets at the same time. But to do that, we need to increase the possibility of multiplexing. So as I told you at the beginning, we have a system that can achieve 7 colors. So the basic approach of multiplexing is when one target is dedicated with one color. But with the color combination approach, we can dedicate 2 colors for one target, and this will increase the number of possibility to increase the number of multiplex that we can reach. So as example, we developed the ECR1 assay that cover up to 18 targets with 17 mutations that were described in the PADA1 consortium. And as you can see here, so for each mutation, we have 2 different colors. And the great thing is that it's really easy to interpret with our software, because here the software will only recognize and calculate the double positive partitions, and all the other partitions will be excluded. So this means that it will decrease also the background noise and the percentage of false positive that could come from single positive partitions, for example. So here, one of our customers also done an experiment very close to what was presented previously with BRAF, NRAS, and KRAS by also doing a sample dilution with 10% of mutation rate and also diluting the mutation in the wild type sample to go up to 0.05% of allele frequency. So as you can see here, we are also very linear in terms of quantification, and also in terms of mutation rate detection, we are around 10%. And for the second experiment, where we diluted the mutation in the wild type, as you can see here, we were able to achieve 0.05%, and we had a very close percentage of the mutation to what it was expected. So this shows again the great sensitivity and the great robustness of the technology, but also the ease of use and the ease of interpretation and a detection of ECR1 mutation in liquid biopsy conditions. So as it's interesting to see this 0.05%, so usually we look to that, we just need to look to the dot plot and to just trust the dot plot. But with the software features we have at Stilla, you are able to select those dot plots and see them in terms of partition and to confirm that they are a true positive partitions and not artifacts or false positive amplification. So this again provides a very high confidence in your data and to be able to achieve this great level of sensitivity. The other example here is comparing also NGS to our technology, especially for low mutation frequency samples. So for the first one, as you can see, the expected mutation rate is 8%, and we were able to detect those mutations with almost a close mutation rate at 10%. But what is really interesting here comparing to NGS is that for very low mutation rates, we do better than NGS, as the NGS was not able to detect those 0.5% of mutations. And we are really confident that are true positive because we have a very high number of positive partitions. And of course, for the wild type, as you can see, we are, of course, concordant. But what is important here to highlight is that the absence of background, because as I said, with the color combination methods, we are able to decrease the background noise. And this, of course, will decrease the number of false positives that could be detected. So here, digital PCR shows better sensitivity than NGS for samples with low allele frequency. In this other example from also one of our users that was using our previous system, which is the NICA 3 system, and using our Sapphire chip with an input volume of microliter, we transferred, or we made a comparison between our new ECR1 assays with the Ruby chip, comparing to this validated methods with the previous technology. So we tested 3 FFP samples with very low concentration, but also 24 cell-free DNA with also a concentration between 0.5 and 3 nanogram per microliter. Some of those samples carry multiple mutations, and here where the panel is very interesting because we can address all those mutations individually. So here is the table of the results. Here we have the samples and the mutation that are expected and obtained with the previous methodology using the Sapphire chip with 25 microliter. And here we have the data that we had with a Ruby chip and with the NIO And as you can see here, even with only 5 microliter of input, we have already a very high concordance with what they developed and they validated with the Sapphire chip. And of course, if we look to 10 microliter or if even 15 microliter, we are still having great concordance. So this shows the level of sensitivity that we can reach with our technology with even very low input volume, as you can see here. And here are some of the concentration level that we tested for those samples. For this last example, a new customer also validated a previous methodology using another digital PCR platform with a duplex drop of assays. And in their validation, they showed that to be able to reach a good and achieve a good sensitivity, they need to run samples in triplicates, which means that they need to have a 60 microliter of input volume. So what we did here is we compared only 15 microliter of Ruby chip with our 18-plex ECR1 assay. And as you can see here, of course, the workflow is again much more easier because we only have one instrument, one consumable, and one pipetting step against multiple instruments with different pipetting steps and different analyzing and PCR times. So here are the results of this comparison. So as you can see here, we have the permutation frequency obtained with the other digital PCR platform compared to what we obtained with our 15 microliter and 18-plex assay. And as you can see here, we achieved a very high concordance score. That means that we are able to achieve very good sensitivity with 4 times less input of precious sample and with very competitive multiplexing capability. And of course, at the end, a very competitive price per reaction because we can target all those kinds of mutations in only one reaction. So this results again was done with our NIO system. So here I will present you the features that we developed with this system. So there are 3 variations of the NIO. The first one is the NIO-E that enable throughput up to 48 sample per run. We can have only one space for one plate, but the great thing here, especially when you want to run multi-panel assays for different kind of cancer or for multiple sample, you can run up to 3 PCR programs at the same time. So this is not only 3 conditions or 3 gradient temperature, it's again 3 completely different PCR that we can run at the same time. For example, you can run BRAF panel with specific thermocycling conditions. In the second chip, you can run completely different thermocycling condition for EGFR, for example, and at the last, another panel that could be also completely different. And with the second version of the NIO, we can run much more throughput as we have 4 free spaces for up to 4 plates. And of course, we can achieve up to 192 samples and 12 PCR program at the same time. And then for the last one, the NIO Plus, we can do a very high throughput with up to 8 plates per run with up to 384 samples per run, and of course multiple PCR program as we have 2 blocks here in the NIO Plus. As I explained, the workflow is really easy. So here we have the Ruby chip So in one PCR plate, we can run 1, 2, or 3 Ruby chips at the same time. And to load the Ruby chip, it's very easy. We just pipette inside the chip and the mix, of course, and the GNA, and then it goes directly to the machine where we will do the partition, the amplification, and the reading step in about 2.5 hours. And at the end, we are able to analyze very high multiplexing data in very easy way with our software, because as you saw previously, for example, with the color combination, it will directly identify the double positive droplets, or depends on the assay, we can also analyze drop-off results very easily. I told you that we are using one consumable. It's again the reality, because we don't use any other consumables like caps or sealing foils or whatever. So we just first the consumable, and then we inject our mix and DNA. The chip is already with oil, so there is no oil manipulation as other digital PCR platform. And again, this consumable is very flexible because it depends on the depth of the sensitivity and the precision. You can choose the level of input volume that you want to have, and also the level of number of partition you want to have, depends on the precision and the sensitivity. So to also be able to achieve this very high number of multiplexing, and also to be able to have this kind of panel, we developed at Stilla a chemistry solution to be able to do that. Because when we developed our system 2 years ago with the 6-color system, the major feedback that came back was, yeah, that's great to have a 6-color system, but it's very difficult to do multiplexing and very high multiplexing panel with the Tachman probes, because it's too expensive and it's too difficult to optimize. So this is why at Stilla we provide a solution that enable a very high multiplexing capabilities with a very competitive price per reaction. So this chemistry is based on two things. The first thing is what we call the flex probe. It's like the Tachman probe, but instead of having a die at 5', we have this flap or tag here that is linked to this probe. And if there is an amplification, the tag will activate what we call a universal reporter that contains the fluorescent dye. And this fluorescent universal reporter is, as it's in his name, it's universal. It means that it's not specific to your target, so it could be used for any panels or any target that you are looking for. Could be mutation, could be methylation, RNA, microbiology, whatever. So with those two approaches, we can use a specific flex probe for the specific target mutation or copy number that we are looking for. We will add the specific tag that correspond to the color of the universal reporter, and we can again use the same set of universal reporters for any panel that we want to develop and we want to use. It's also a modular approach that enable you to build your own panel, depends of the target that you are looking for, because again we will use the same universal reporters. So to have some examples of those assays that are already on our catalog, you can look in our website to find if the targets that you are looking for are already developed by our team. And you can also use this barcode to directly reach the website. If the panel that you are looking for is not in our catalog, we are more than happy to develop your assay and your panel for you, depends on the target and the panel that you want. So we can develop it for mid, for also starting from low, mid, and high multiplexing. Depends again on your need. If it's, for example, for screening or is it for individual targeting. And of course, with our service, we can also provide the positive control that will help you for your optimization and for your testings. So I hope that after this presentation, I convinced you that with our technology, you can already start using next generation digital PCR to enable high sensitivity analysis for low concentration sample and target. And of course, to have the maximum information analysis from precious sample using high multiplexing capabilities. Of course, all this again with a very easy workflow with minimal hands-on to also reduce experimental variability. With our flexible throughput, you can adapt the technology and also, again, the level of the sensitivity and precision you want to reach. And this, again, with a time and cost saving for target screening compared to other technology or other digital PCR system. Those are some feedback that I want to share with you from our users that tested our new NIO system. And also, I want to say a big thank you to our customers and users and partners that were very happy to share their data with us for this presentation today. I want also to share with you this great news that Stilla technology was chosen among the 100 best French tech and companies, and we will present our NIO digital PCR in the coming days in the Palais de l'Élysée. So we are really proud about it. So please keep in touch and follow us to see this event. Thank you for your attention and hope you enjoyed the session. And now we are open for answering your questions.
Thank you very much, Sahbi, for the interesting talk. So now we open the forum for some questions. You can post your question by going into the tab with the question mark item on the top. So I'll be modulating the questions now. I see some of them coming up. To start with, what kind of liquid biopsy samples did you use for the experiments you described?
So for some of the data that customers shared with us, there were plasma samples.
There's another question saying, if we have validated assays on the NICA system, can we transfer them to NIO?
Yes, of course. For the one example I showed with the Eugène Marquis Center where they developed their assay. Again, we tested their assay, but also our assay on the NIO. And of course, the transfer is very easy to do.
Perfect. So I see another question coming up. It's more about the question of, you were mentioning that you can run multiple panels within the same run. Can you do the multiple panels within the same chip?
Yes. So of course it will be depends on the thermocycling conditions, but also one thing that is interesting about the universal reporter is that it makes easy to have panels with close thermocycling conditions. So this will enable to run multiple panels in the same chip. Of course, it doesn't mean that we will not run through some optimizations, but it's again more feasible with the universal reporters than with the TACMAN probes.
Yeah, so actually on that question about the Crystal Universal Reporter, there's a question about the Crystal probe assays. How do they limit the optimization regarding dimer concentrations, since their primers are intact and tag probe are so specific to the target?
So I will start from the end. So the tag is not specific to the target. The tag is specific to the color of the universal reporter. And to get back to the first part of the question, the in silico design of an crystal assay using universal reporters follow the same rules as the design optimization like for TACMAN. The only thing here in which it lead to an easy optimization is that the universal reporters that have fluorescent dye is already optimized at Stilla. Because if you remember, or maybe we all ran some TACMAN optimization in the past, so the most difficult part is to optimize this fluorescence multiplexing with the TACMAN assays using different dyes. We need to find the right dyes, the right quenchers, and the right wavelengths, etc., etc. And this part is already validated and optimized within Stilla for our universal reporter. So this is why the optimization of the assays using this chemistry is easier than TACMAN probes.
So some more questions around the assays design and the. So there we have some feedback saying, "Hey, great stuff." So thank you very much for that. So the following question is: to gain specificity for the mutations, do the PCR primers need to be allele specific?
Yes. So I mean, not the primer. It would be more on the broad. I mean, it really depends because there are different strategy for the in silico design. So basically, we can use, for example, LNAs to increase the sensitivity and to be able to distinguish between allele A and allele B. We can use also the allele-specific strategy, but again, it will really depend on the target that we are looking for. But it's follow again the same rules as allele detection with TACMAN probes.
A follow-up question is: when you showed the sensitivity results, there were 2 out of 3 positive in the wild-type samples, even higher than lower concentrations of positive samples. How do you deal with false positive results?
Of course, so those data were just really primary data showing the level of sensitivity that we can reach. But again, to deal with false positive, the best and the well-described methodology for that is to calculate the LOB and the LOD of each assays as described for the digital PCR MACI guidelines. So this is the easy, or let's say the best way at first. The second one that could be considered as false positive, and this we can and sometimes go across it, is when there is, for example, artifacts in the partitions. And there, of course, the software will exclude them. And also we have this QC that we are able to see using the feature that we have in our software called Explore Crystal that enable you to select the partition and to see this positive partition inside the QC image and to see if it's a true positive with an homogeneous fluorescent signal inside the partition, or is it a false positive or for an artifact. So this is another way.
So then there's another question. Thank you so much for all these questions coming up about the primer design. How do you go about primer dimer optimization?
Again, it's really like I would say qPCR or basic digital PCR design. So this is also what is really good, that we again follow the already described rule for assay design. So for primer dimers, for example, we can first check them within silico design, and of course afterwards we run several specificity experiment to see if we have extra population. For example, this will show that if the primers are specific or not. If we have dimers, also we can check them on qPCR with the usual methods. So we again follow the same rules as what it was described for the basic assays.
So next question is the main advantage of the Crystal Universal Reporter just to save costs and time? Having the extra step of the flat cleavage seems to make the reaction more complicated and may affect reaction sensitivity.
So no, because first of all, about so it's not only about time and cost, it's also about sensitivity, because as I showed, for example, for the ECR1 18-plex, using the color combination method. So basically, if we go back also to the origin of why we are thinking about those universal reporters, to make color combination easier and affordable for everyone. So to be able to do that, we developed this universal reporters. And by using this color combination method, again, we will only visualize and select the double positive partitions for the specific target. This means that all the other partitions that don't have this double dyes will be excluded from the analysis and will decrease the background noise, which increase the sensitivity, which means that we will have a very low LOB. So if there is one partition that is positive, we will be able to see it. This is the first thing. And the second thing, I forget about it. It was about the specificity, the sensitivity, and
yes, and how can it affect if it will be there make the reaction?
Yeah, no, not at all, because it's exactly the same PCR condition. So it doesn't mean that we have an extra step for that for the tag or flap cleavage and activation. It's done within the same step of annealing and extension. So there is no extra step or time. It's really, I won't say transparent and invisible for the user. So when you use the PCR condition, it's like TACMAN PCR conditions.
And then I can add also a usability feature about the Crystal Universal Reporters is the fact that the fluorescent molecule that is non-specific to the target but can be used across different panel mutations will be the same. Therefore, stock management across different laboratories or different experimental setups is greatly simplified, as you have the single fluorescent molecule that could be used across all the Crystal Flex bulk panels. So that is a great advantage that our customers have appreciated, especially when you start developing more and more panels with the technology. Then stock management becomes quite easier.
Exactly.
So we have many questions coming up. We will probably tackle those via email. So don't worry, we'll address those questions as we follow up after the seminar. So I wanted to thank Sahbi for the time today and you for your participation to this seminar. If you have any other questions regarding the capabilities of the NIO digital PCR systems, color combinations, or the Crystal Flex Probe assays, don't hesitate to contact us or visit our website. A recording of the webinar will be sent in case you want to rewatch afterwards. So with that, I will close this session and thank you very much for your participation.
Thank you very much. Bye.